Usually when we talk about a medical diagnosis, there's this underlying expectation of precision.
Right, like it's engineering or something.
Exactly. Feels like engineering. You break your arm, the x-ray shows a jagged white line, and the doctor just points to the screen and says, there's the problem.
It's very binary, which is honestly incredibly comforting for a patient. Our human brains just want things to be visible. We want it easily categorized, broken or not broken.
Yeah, but step into the world of oncology, and specifically gastric or stomach cancer, and suddenly that really neat binary x-ray machine just shatters. We are looking at a diagnostic landscape that is, well, it's exceptionally murky.
Murky is the perfect word for it, yeah.
Because think about this. You can have two patients present with the exact same tumor size. They undergo the exact same surgery. They receive the exact same chemotherapy regimen. And yet
one survives and thrives, and the other's disease just progresses rapidly.
Right. Why? Which is the mission of our deep dive today. We're trying to crack the code of predicting how stomach cancer will behave, and we're doing this by looking at a groundbreaking 2026 multi-center retrospective study.
And this study is wild because it points to a fundamental flaw in how we currently categorize this disease. The global standard right now for making these literal life or death decisions is the TNM staging system.
TNM, so that's tumor node metastasis.
Exactly. It measures how deep the primary tumor has actually invaded the stomach wall, how many local lymph nodes have cancer cells in them, and whether it has spread to distant organs.
Okay.
But the issue is it provides a structural snapshot. It completely misses the underlying biological behavior that's actually driving that structure.
I always think of TNM staging like judging a novel strictly by its physical dimensions and its page count.
Oh, that's a great way to put it.
Right. Because it tells you the size of the book, sure, but it reveals absolutely nothing about the actual story inside. It doesn't tell you how aggressive the characters are or, what kind of plot twists are hiding in the next chapter.
That analogy totally holds up, though I'd probably add that TNM is basically just reading the back cover blurb.
Yeah.
The actual narrative of the tumor, the real story, is written in its genetics, its molecular makeup. Now, clinical oncology obviously does have tools to read that deeper story.
We have the molecular tests.
Right. We run advanced molecular tests looking for very specific markers, like microsatellite instability or PD-L1 expression. We're basically looking for specific gene mutations that tell us exactly how the tumor operates.
But the problem, and this is based on the research we're diving into today, is that those molecular tests are a massive bottleneck.
A huge bottleneck. They are incredibly expensive for one. They require pristine tissue samples, and they can take weeks to process.
And for a patient sitting there waiting to hear if they need toxic chemotherapy, time and money are critical barriers.
Which is why the holy grail of oncology right now is finding a method to extract that deep molecular data without actually running the expensive molecular tests.
And this 2026 study claims to have found a shortcut. They're using a novel artificial intelligence framework to analyze something called Pathomics.
Right, Pathomics. They are extracting hidden predictive data from standard, incredibly cheap pathology slides.
So let's talk about the raw material first.
Yeah.
This study relies entirely on H&E slides, hematoxylin and eosin.
Yeah. Anyone who has ever watched a medical drama on TV has seen these. They're those little glass slides with the pink and blue tissue smears that doctors look at under a microscope.
Okay, but what is the pink and blue actually doing?
Well, the pink and blue is actually a really specific chemical reaction. It's been the bedrock of pathology for over 100 years. So hematoxylin is a basic dye, and it binds to acidic structures in the cell.
Like the DNA.
Exactly, the DNA and RNA inside the nucleus, and it turns them this dark purplish blue color. Eosin, on the other hand, is an acidic dye that binds to basic structures, primarily the proteins in the cytoplasm and the extracellular matrix, and that stains them various shades of pink.
So it basically gives you a chemical map of the cell's density and its physical structure. And because it's a 100-year-old technique, it literally costs pennies. Every hospital on Earth can do it.
Exactly. It's universal.
But I'm struggling with the central premise here. We have had the world's most brilliant human pathologists staring through microscopes at these exact same pink and blue smears for a century.
Right.
Are you telling me a computer algorithm is just magically seeing things that top-tier doctors are completely blind to?
It's not magic at all. It's mathematics. When a human pathologist looks at that slide, they recognize macro patterns based on decades of training. They look for a misshapen cell or maybe a dense cluster of tissue that just shouldn't be there.
They're doing visual pattern recognition.
Right. But the AI, specifically through deep learning in this Pathomics approach, it analyzes the microscopic pixel-level spatial relationships across millions of data points simultaneously.
Why?
It's looking at the raw mathematical gradients of those pinks and blues and finding geometric correlations that are, frankly, literally too complex for the human visual cortex to process.
So it's seeing the underlying code of the image, not just the picture itself, which naturally brings up the medical community's absolute greatest fear regarding AI.
The black box problem.
Exactly, the black box.
It is the single highest hurdle for clinical integration right now. I mean, an AI model might boast a 95% accuracy rate in predicting cancer survival, but if it operates as a black box, meaning data goes in, a prediction comes out, and the machine cannot explain how it arrived at that conclusion, doctors will flat out reject it.
Because you can't ethically prescribe a highly toxic chemotherapy regimen on a machine that just says, trust me, bro.
Exactly. You cannot do that.
So just to recap this first part for you listening, the standard T&M staging just isn't enough for predicting gastric cancer outcomes. The advanced molecular tests are too expensive and complex. H&E slides, the pink and blue ones, are cheap and everywhere. And the massive challenge here is making an AI read these cheap slides without it being a mysterious black box. Think about a time you had to trust a complex algorithm, like a credit score or the feed on your social media. Would you trust it with your literal medical treatment if it couldn't explain its reasoning?
I definitely wouldn't, and that is the tension this research attempts to resolve. They built an AI model that not only reads the microscopic data, but forcefully requires the algorithm to show its work.
And they call this model MSGMIL?
Yes. Multi-scale graph neural network with gated attention mechanism for multi-instance learning.
Okay, wow. We absolutely have to break that down. Multi-scale graph neural network with gated attention.
I know, it's a mouthful.
Let's start with the input, because we aren't just feeding a small JPEG from an iPhone into a computer, right?
Not at all. A whole slide image, or WSI, of a tumor is a 10 gigapixel file.
10 gigapixels. That is massive.
It's unbelievably huge. Feeding a 10 gigapixel image directly into a neural network would just instantly crash the system. There is simply too much data. So the very first step in the MSGMIL architecture is chopping that massive image up into thousands of tiny, manageable patches.
How small are we talking?
Each patch is 512 by 512 pixels. But here's the clever part. They extract these patches at two different magnifications, a 20x zoom and a 10x zoom.
Okay. That is the multi-scale component of the name.
Exactly, multi-scale.
It's kind of similar to analyzing a city using a satellite map. The 20x zoom is like Street View. You're looking at the individual houses, which in this case are the specific tumor cells and their immediate nuclei.
Right.
And the 10x zoom is the zoomed out city grid showing you the neighborhood, the actual structural layout of the tissue.
That is a very apt comparison. So once the image is chopped into those multi-scale patches, the model uses something called a SWIN transformer.
SWIN transformer, what does that do?
Transformers are state-of-the-art feature extractors. They basically scan the pixel gradients in every single patch to identify fundamental textures, edges, and densities.
Okay.
But identifying individual patches in isolation is completely useless. A cancer cell does not exist in a vacuum. You know, its behavior is heavily dictated by its environment.
The tumor microenvironment.
Precisely. And this is where the graph neural network, or the GNN part of the acronym, comes into play.
Okay, so the GNN maps the spatial relationships.
Yes. It takes all those individual chopped up patches and mathematically calculates how they relate to one another geographically. It maps out, say, how a malignant epithelial cell interacts with a neighboring immune cell, or how close it is to a blood vessel.
So the algorithm is essentially building a social network for the tumor.
That's exactly it.
It's figuring out who is talking to whom and which cells are actively influencing the surrounding tissue.
But there is a catch. A lot of that 10 gigapixel image is just going to be a blank glass slide, or maybe perfectly healthy tissue that has absolutely nothing to do with the cancer.
Which introduces massive amounts of noise.
Massive amounts. If the neural network treats the empty glass and the highly aggressive tumor core with equal computational weight, the predictive signal just gets completely drowned out. That's why the architecture includes a gated attention mechanism.
Okay, so gated attention. It functions like a dynamic filter.
Exactly. It assigns a probability weight to every single patch. The algorithm basically learns over time which structural neighborhoods contain the actual predictive value. The gated attention mechanism forces the AI to focus all of its computational power on those critical hotspots.
The fires, basically.
Right, fires. And it actively ignores the empty fields, the irrelevant background noise.
Okay, so let me get this straight. The model chops up the multi-scale map. The transformer extracts the tiny textures. The graph neural network maps the social connections between the cells. And the attention filter removes all the noise.
You got it.
When all of that insane math is finally finished, what is the actual output? What does the doctor see?
The model feeds all of those extracted, spatially mapped features into something called a Lasso-Cox regression analysis.
Lasso.
Yeah, LASSO stands for Least Absolute Shrinkage and Selection Operator. It's basically a statistical technique that penalizes the algorithm for being too complex.
How so?
It ruthlessly forces the mathematical weight of the less important features down to absolute zero. It shrinks the data pool until only the most powerful, undeniably predictive features remain.
It's trimming the fat until you only have the MVP variables left.
Exactly. It narrowed thousands and thousands of variables down to just 11 specific features. And by combining the weight of those 11 features, it calculates a single actionable number. The Pathomics signature of gastric cancer, or the PSGC score.
The PSGC score.
Yes, and the researchers analyzed the survival data and found an optimal mathematical cutoff at -1.37.
Meaning if a patient's PSGC score is mathematically higher than -1.37, they are classified as high risk. And if it's below that line, they are low risk. Okay, let's do a quick recap of that beast of a mechanism. MSGMIL chops images into multi-scale patches. Transformers extract the features. GNNs map the spatial relationships, the social network. Attention mechanisms filter out the noise. And it all results in a PSGC score with a cutoff of monocle 1.37, determining high versus low risk.
Spot on.
So for you listening, if you were building a social network of a tumor, which cells do you think would be the most important influencers? The cancer cells themselves or the surrounding healthy cells? It's something to think about.
Definitely.
But a fancy algorithm generating a score in a lab means nothing until you prove it works on real human beings across different populations.
And the validation scale for the study was truly staggering. They didn't just test this on a handful of local patients. They tested the PSGC score on 3,138 patients.
Wow, over 3,000.
Across 5 completely different hospital cohorts to ensure demographic and clinical variety. They even validated it against the TCGA STAD dataset.
Right, the Cancer Genome Atlas Stomach Adenocarcinoma Collection. That is a massive, globally recognized public database holding deep molecular and clinical data for cancer patients.
Exactly. By using the TCGA data, they proved that AI isn't just, memorizing the specific slide preparation quirks of one single hospital. It works universally.
It's robust.
Very robust. And the results across all those cohorts showed that the PSGC score independently predicted overall survival. But the researchers didn't stop there. They actually pitted their AI against the old standard T&M staging system we talked about earlier.
Oh, wow. A head-to-head.
Even better, they combined them. When they merged the AI's PSGC score with the traditional T&M staging, creating a composite model, the predictive accuracy just skyrocketed. They measured this using something called the C-index, or concordance index.
I want to make sure we understand the C-index, because it comes up a lot of these deep medical data states. It's basically a ranking metric, right? So if you take 2 random patients from the study, and one died sooner than the other, the model should have given the worst score to the patient who died first. The C-index measures how often the model gets that ranking correct.
That is exactly how it works. A C-index of 0.5 is basically a coin flip. 1.0 is absolute perfect prediction.
Right.
The composite model integrating the AI scored significantly higher on the C-index than the T&M system alone. It also vastly outperformed the standard system on net reclassification improvement, or NRI, and the IDI scores.
Which basically means it successfully took patients that the old system had incorrectly labeled as low risk and properly reclassified them as high risk before their disease progressed.
Exactly. It fixed the blind spots.
So if we have this incredibly accurate AI-enhanced survival score, how does it actually alter the timeline for a patient? Because survival prediction isn't just about, waiting for the clock to run out. It's about deploying the right weapons.
This brings us to the most critical clinical application of the entire study. The adjuvant chemotherapy dilemma for stage 2 and stage 3 gastric cancer.
Right.
After a surgeon goes in and removes the primary tumor, the standard protocol often involves aggressive post-operative chemotherapy regimens. things like XCLOX or SOX regimens.
And these are intense systemic treatments. We're talking about regimens that cause severe nausea, peripheral neuropathy, total immune system suppression.
It is a brutal, brutal process for the human body to endure. And the really tragic reality of standard TNM staging is that doctors often simply cannot predict which specific patients will actually benefit from that toxicity.
And which patients will suffer the horrific side effects without gaining a single extra day of life.
Exactly.
That is arguably the darkest part of oncology, enduring the poison when the tumor is already resistant to it. So can this AI score, generated from a simple pink and blue slide, actually predict chemical resistance?
The data is definitive, yes. The study revealed that patients identified as having a high PSGC score demonstrated a massive statistically significant survival benefit from receiving that postoperative chemotherapy. The chemo worked for them. It actively hunted down the microscopic disease left behind. However, the patients with a low PSGC score experienced almost no significant survival enhancement from the chemotherapy.
I need to pause here because the implication of that is just staggering. An algorithm looking at the geometric arrangement of pixels on a photograph can tell us if a patient's biology will respond to highly toxic intravenous chemicals.
It sounds like science fiction, I know.
It feels like looking at a satellite image of a dry farm and predicting exactly which brand of fertilizer the soil will absorb best. How does an image predict a chemical reaction?
Because the image is not just a picture. It is the physical macro-level manifestation of the tumor's microbiology.
Okay, explain that.
The AI isn't predicting a chemical reaction in a vacuum. It is recognizing how aggressively the tumor is building its cellular defenses. A tumor that is rapidly mutating and building chaotic structures leaves a literal visual footprint. And that structural chaos is exactly what makes it vulnerable to chemotherapy, because chemo specifically targets rapidly dividing cells.
Oh, it's reading the tumor's architectural weak points.
Precisely. And they found this predictive power extends to immunotherapy as well.
Wait, really?
Yes. Patients with a high PSGC score were significantly more likely to achieve a partial response to anti-PD-1 immunotherapy. But the low PSGC patients faced progressive disease despite getting the treatment.
Wow.
This completely moves the needle from generic, stage-based protocols to true individualized precision medicine.
Right, because we can spare the low PSGC patients from the agonizing side effects of a drug that won't help them anyway, and immediately pivot them to alternative trials.
Exactly.
So quick recap of segment 3. The PSGC was validated on over 3,000 patients globally. It perfectly complements TNM staging. High PSGC means a patient will likely benefit from chemo and immunotherapy. Low PSGC means those treatments might not extend survival.
That's the summary, yeah.
So think about it. How would knowing this change the conversation you have with a doctor if you or a loved one were facing months of harsh chemotherapy? It changes everything.
It completely changes the risk-benefit analysis.
But none of this matters if we can't solve that black box problem we talked about earlier. The researchers had to prove why the AI was making these high and low risk categorizations.
And they did. To rip open the black box, the researchers deployed two really powerful interpretability algorithms, SAP and GradCam.
SAP and GradCam. Let's start with SAP.
SAP operates somewhat like a forensic accountant. You recall the model used a lasso regression to narrow thousands of variables down to just 11 features.
Yeah, the MVP variables.
Right. SHAP analyzes that final model and assigns a definitive numerical weight to each of those 11 features, showing exactly how much influence each one had on the final score.
So it literally shows you the math. And what was the MVP? Did one stand out?
Oh, absolutely. Feature 17 was the undisputed heavyweight. It drove the high-risk predictions more than any other variable.
Feature 17.
Yes. But SHAP only gives you the numerical accounting. It's just numbers. To translate that back into human biology, they had to use GradCam.
Which stands for Gradient-Weighted Class Activation Mapping. What does that actually look like?
Well, GradCam traces the mathematical gradients backward from the final prediction to the original image. Think of it as applying thermal goggles to the AI's visual cortex.
Thermal goggles, okay.
It generates heat maps overlaying the original tissue slide, literally highlighting the exact microscopic regions in glowing red that the algorithm stared at the longest to make its high-risk determination.
So they essentially printed out these red-hot heat maps, handed them to top-tier human pathologists and asked, hey, what biological structures are sitting right under these red pixels?
Exactly. And to make sense of the thousands of heat maps, they used high-dimensional data crushers, algorithms like k-means clustering and TSNE to group visually similar AI outcomes together.
And what did they find?
They found incredibly consistent biological patterns. For the high-risk bad prognosis patients, which were grouped into clusters 6 and 7, the AI's thermal goggles were locked onto areas showing high cellularity, nuclear pleomorphism, and tumor-stroma fibrosis.
Let's unpack those. Nuclear pleomorphism. That's when the nuclei of the cells, the control centers, are wildly mutated, right?
Yes, they vary heavily in size and shape.
It's a classic hallmark of aggressive, chaotic division. And what about the tumor-stroma fibrosis?
Well, the stroma is the connective scaffolding tissue surrounding the tumor, and fibrosis is the formation of dense scar tissue. So the AI recognized that the most dangerous tumors actively build a thick, fibrotic wall of scar tissue around themselves.
Like a fortress.
Exactly. This wall acts as a physical shield, literally preventing the body's immune T cells and the chemotherapy drugs from penetrating the tumor core.
The AI is literally seeing the tumor building its fortress.
Yeah.
That is incredible. And what was it looking at for the low-risk, good prognosis patients?
Yeah.
The ones in cluster 0 and 2?
For the good prognosis, the AI focused heavily on areas showing intraepithelial neoplasia and intestinal epithelial metaplasia.
Okay, translate that for us.
These are specific cellular changes frequently associated with chronic H. pylori bacterial infections in the stomach.
H. pylori.
Right. Interestingly, clinical data has long shown that gastric tumors associated with H. pylori infections often have slightly better survival rates compared to non-infectious, spontaneously mutating tumors.
Wait, so the AI independently figured out that clinical correlation just by analyzing the pixel gradients?
Yes. It essentially learned a century of pathology from scratch, just from the math.
That is mind-blowing. But the researchers went one step deeper, didn't they? Because finding a visual correlation is great, but they wanted to prove that these visual patterns are directly linked to the actual genetic mutations happening inside the patient's DNA.
Right, and this is arguably the most profound part of the study. They cross-referenced the AI's visual risk scores with the actual RNA sequencing data from the tumors. They used gene set enrichment analysis, or GSEA.
GSEA.
Yes, it looks at entire networks of genes to see which biological pathways are turned on or turned off.
So they are bridging the architectural photograph with the raw genetic code.
And the correlation was flawless. They discovered that tumors assigned a high PSGC score by the AI, the aggressive high-risk ones, had hyperactive EMT pathways in their genetics.
EMT, epithelial mesenchymal transition. Let's clarify that for anyone fuzzy on cellular biology. Epithelial cells are the standard cells lining your stomach, right? They act like bricks in a wall. They're anchored together by adhesion molecules, keeping everything tightly in place.
Yes. While mesenchymal cells, on the other hand, are mobile. They don't stick together.
Right.
So when a tumor activates the EMT pathway, it is effectively instructing its cells to dissolve their anchors. The stationary bricks mutate into rogue, highly mobile agents that can break away from the primary tumor, slip into the bloodstream, and invade other organs.
It is the fundamental genetic mechanism of metastasis.
Yes, and the AI could see that happening without sequencing a single strand of DNA. It saw the physical loosening of the tissue structure, the irregular shapes that occur when a cell drops its anchors.
That's wow.
It gets better. The genetic analysis also revealed heavy stromal cell infiltration in the high-risk patients. Using a tool called Excel, they found that the tumor microenvironment was flooded with endothelial cells.
Endothelial cells. Those are the cells responsible for angiogenesis, right? The construction of new blood vessels.
Exactly. The tumor is genetically hijacking the body, forcing it to build new vascular pipelines to feed the cancer's rapid growth. The AI couldn't read the RNA, but it could physically see the microscopic vascular networks being constructed in the stroma.
It's like tracking a predator by analyzing his footprints in the broken branches in the forest, rather than needing to actually catch it and draw its blood. The macrostructural image is entirely dictated by the microgenetics.
Which completely validates this entire field of pathomics. We no longer have to view standard pathology slides as just structural snapshots. They are incredibly dense topographical maps of the tumor's genetic operating system.
Okay, let's recap this final segment before we wrap up. SAP and GradCam heat maps open the black box. High risk equals tumor fibrosis and deformed nuclei. Low risk equals intestinal metaplasia. And the visual patterns perfectly correlate with genetic pathways like EMT and tumor microenvironment infiltration.
Perfectly summarized.
So, to you listening, turns out the AI was learning the visual language of our DNA. What stands out to you about the fact that our genetic pathways leave visible physical footprints in our tissues?
It's a lot to process.
It really is. We have covered a massive paradigm shift today. We took a 100-year-old chemical pink and blue tissue staining technique. We fed it through a multi-scale neural network that maps the spatial social network of mutated cells.
We used gated attention to filter out the noise.
Right, and lasso regression to distill it into a single score. And that score not only predicts survival better than our current global standard, but it dictates whether a patient will actually respond to highly toxic chemotherapy.
And by opening the black box, the researchers proved the AI isn't just guessing. It is actively reading the physical footprints of genetic metastasis.
Which leaves us with a deeply provocative thought to consider. If an algorithm can look at a cheap, basic H&E slide from a stomach tumor and accurately deduce its complex genetic mutations, its immune infiltration, and its future drug resistance.
What else is hiding in the data?
Exactly. What other hidden secrets are lying dormant in the billions of routine medical images, the chest x-rays, the MRIs, the basic blood smears currently sitting in dusty hospital server archives all over the planet right now? Are we sitting on the cure to diseases, just waiting for the right algorithm to look at the data we already have?
The data's already there. We just finally have the lens required to read it.
Thanks for joining us on this deep dive. Keep questioning the obvious, and we'll see you next time.